Hey class, hope you guys survived the winter storm last night. It was amazing waking up this morning to so much snow. Super cool. Well, today we're gonna learn about human reproduction and development and tie that in along with meiosis to inheritance, basically how you pass on your genes and how those come together to form so many diverse individuals. So, before we get into human reproduction, let's talk about reproduction in general and why sexual reproduction is advantageous. So, here you have two organisms, an amoeba and a cat, right? And an amoeba is an example of an organism that undergoes sexual reproduction, and this is going to produce genetically identical organisms. So the amoeba here, you can see that it's a single-celled organism and it divides via mitosis and produces two genetically identical amoebas. And sexual reproduction is where you produce genetically diverse organisms by combining two different organisms' genetic content. So male and female coming together. And you can see here the cat, her offspring are genetically diverse, right? Even by the fur color. And genetic diversity, as we've talked about, is a good thing. That is good news for the offspring because being diverse helps survive environmental pressures such as predators, pathogens, drought, high temperatures or low temperatures, etc. In other words, genetic diversity allows for adaptation to different environmental pressures, which will eventually allow the animal to survive, right? That's survival of the fittest, that idea, which we'll talk about towards the end of this class. There are three main mechanisms that create genetic diversity. The first one is through mutations, where a mutation in the DNA sequence of a gene will eventually result in a mutation in the amino acid sequence of a protein, and that can create different versions of genes called alleles. And this is the first and most foundational level of genetic diversity. Then we also looked at meiosis and how through crossing over and independent assortment, meiosis can generate genetic diversity. So, for example, males and females each could make 8 billion or more gametes, sperm or eggs, and each one of them is going to be different from the other. No two will be alike. So, literally, you could have 8 billion children and none of them would look the same. And then the last mechanism for genetic diversity creating that is sexual reproduction, where you have 8 million combinations of chromosomes in the sperm, 8 million combinations of chromosomes in the egg, and those two can come together and form 70 trillion, can come together in 70 trillion combinations, right? So incredible genetic diversity from meiosis and sexual reproduction. All right, so let's look at how gametes are made. Gametes are simply sex cells, and meiosis is a cell division process by which we make our gametes, whether it be sperm or eggs. So when a haploid sperm, which a haploid cell is a cell that has half the amount of the normal genetic or normal chromosomal content, so in this case that's 23 for humans. So when you have a haploid sperm and a haploid egg and they fuse together through fertilization, we end up making a diploid zygote. So a diploid cell is a cell that has two sets of chromosomes, 23 and 23, right? So a set from dad and a set from mom, and that makes 46 chromosomes. And now this diploid cell, this diploid zygote, can begin dividing and becoming an actual organism through mitosis, right? So the way you go from one cell to 37 trillion cells, or how many ever cells you have when you're born, that is a process of mitosis. Meiosis is a process by which, as an adult, you make your sex cells or your gametes. So now let's look at how males and females make their gametes. So males make their gametes, their haploid sperm, via a process called spermatogenesis. And here we have an image of the testicle, which is basically the sperm factory of the male. And in the testicle, you have these tubules, which are like little tubes where the sperm is being made. And you have a ton of this. It's just all coiled up and packaged in the testicle. And as the sperm is released, it's collected in the epididymis and then released into the vas deferens so it can exit the body. So this seminiferous tubule, if we look at a cross-section of it, we can see that it's a bunch of cells. It's a tube. It has a center, hollow center there in blue. And you have immature sperm cells near the outside layer that become mature sperm by the end of that cross-section there, where from the outer layer to the inside of the tube, and then the sperm are released into the tubule. So it can go into the epididymis and out the vas deferens. All right, so let's look at that little cross-section of the cross-section. So in the outer layer, you have the immature sperm cells, and these immature sperm cells are diploid, and they actually are dividing by mitosis so that they can make a constant supply of sperm. So they'll divide through mitosis and produce a primary spermatocyte, which is that second cell there that has the 2 chromosomes that are in that characteristic X shape. So this primary spermatocyte is a diploid cell. For example, we're only looking at one chromosome here. Say this is chromosome 1, and you can see that you have one from your mom in red and one from your dad in blue, right? And they're replicated. So this primary spermatocyte then is going to begin to mature into a sperm cell. And it does this through meiosis, which means it'll go through 2 rounds of cell division and produce haploid sperm. So this primary spermatocyte, which is a diploid cell, has 46 replicated chromosomes. So basically it's got double the normal content actually. So normally you have 46 chromosomes. These are 46 duplicated chromosomes, which is why they're in that X shape. It'll go through the first round of cell division, and these cells will have 23 duplicated chromosomes. They'll go through another round of cell division, and those duplicated chromosomes are going to split in half basically, and you're going to end up with 4 haploid spermatids, which then mature into sperm or spermatozoa. And these then are released into the tubule and exit the body, or collect in the epididymis and then exit the body. And this is a continuous process. So as the cell there on the outer layer, the very top of this last diagram, continues to divide through mitosis, those primary spermatocytes continue to mature into sperm cells. So now let's look at how females produce their gametes. And they do this via a process called oogenesis. And this is similar to how males make sperm, where you go through 2 rounds of meiosis, right? You go through 2 rounds of cell division, 1 round of meiosis. And the interesting thing about oogenesis, how females make their eggs, is you're actually born, all of you women listening to this, with all of the eggs that you will ever release in the form of 1 to 2 million primary oocytes. So just like you have primary spermatocytes, which are immature sperm, you have primary oocytes, which are immature eggs that are going to go through meiosis to become a mature egg. So to the left of the figure, you have the ovary, which is the female egg factory, and that's connected to the uterus by the oviduct. And the uterus is where the egg in the sperm, or the fertilized egg, is going to attach to the walls and then begin human development. So let's look at the ovary, and here it's a diagram, simplified diagram, of the ovarian cycle, how you go from a immature primary oocyte to a secondary oocyte. So we're going to go through 4 steps, again, very simplified diagram of the process, 4 steps of how we make an egg that's ready for fertilization. So number one, you have there a primary follicle, which is just a group of cells that surround the primary oocyte. And so they form, it's like a little oocyte compartment, if you will. It keeps it nice and safe. And it also has these cells, these light orange cells, that release all of these hormones. So your brain is actually releasing hormones, which is telling these follicles to begin to mature, the oocyte. So they begin to release estrogen and begin to grow. These cells begin to divide, the orange cells, and begin to release more fluid, which then begins to form a little pocket there and forms a secondary follicle cell and releases more estrogen and progesterone. So all these hormones that are making women make you feel like you're going crazy, and that's the menstrual cycle. And this secondary follicle is going to continue to grow, become another phase of follicle. But basically, this is, you can tell that that follicle is grown and it's kind of bulging, pressing against the side of the ovary, and it's going to release some enzymes that are going to poke a hole through the ovarian wall and releases the secondary oocyte. And at this stage, this is the process that's known as ovulation. And it's taken basically 14 days or so from the primary oocyte till this release of the secondary oocyte. And we're not going to talk about the rest of it in depth. You can read this in your textbook if you want, but you can see that there the follicle that is now left behind turns into what's known as the corpus luteum. And these are a bunch of cells still in the corpus luteum that are still releasing a bunch of hormones. So these are the granulosa cells, the cells that have been releasing hormones all along this ovarian cycle. And they're going to continue to release hormones that are going to prepare the uterus to receive a fertilized egg, if that were to happen, so that that egg can implant and then begin to develop into an embryo and then into a fetus. But if the egg doesn't get fertilized, then the corpus luteum eventually is going to degrade, stop releasing hormones, and the lining of the uterus that had been developing to receive the egg, the fertilized egg, it's going to fall off, which is why women have a period. And again, you can read about this more in depth in the textbook. Now, something else that's interesting about oogenesis is that you go through only one round of cell division from a primary oocyte to a secondary oocyte. So you don't complete meiosis. You actually don't complete it until that egg is fertilized. And in meiosis 1, so you go through one round of cell division, meiosis 1, and you end up doing unequal cytokinesis. Remember, cytokinesis is the step at which the two dividing cells finally separate. And so unequal cytokinesis is, instead of producing two equal sized cells, you produce two unequal sized cells. So here you have the primary oocyte, and this is a diploid cell. It has 46 chromosomes. And you go through meiosis 1 and cytokinesis, and you can see that the two resulting cells are very different. So you end up with a secondary oocyte, which is fat and happy, but you also end up with something called a polar body, which also has a nucleus, but it has taken absolutely none of all of the good stuff, the cytoplasm. So that's actually going to degrade and die off eventually. And the secondary oocyte is going to continue. And it does this because that way, even though the cell is dividing, the oocyte is left with all of the good stuff. The secondary oocyte is left with all of the good stuff. And so this is a secondary oocyte that's being released during ovulation, and it's actually not going to go through that second part of meiosis until sperm fertilizes the oocyte. So once fertilization happens, that egg is going to go through another round of division. That secondary oocyte is going to go through another round of division and release another polar body, the second polar body. And then after that, the nuclei of the sperm and the egg are going to fuse. So let's look at how that happens. So fertilization is a step at which the sperm and the egg fuse together. And here in the diagram, you can see that first a sperm will penetrate the follicle cells. So there's actually a layer of cells on that secondary oocyte that's released. And the sperm has to penetrate that, and then it has to penetrate this region called the zona pellucida. And it's just got a lot of proteins and carbohydrates, and it's like a protective coating. So the sperm is going to have to penetrate that. And once it does, in step 3, you see that it's going to release acrosomal enzymes, which are just enzymes that are going to change that zona pellucida and harden it. And it does this so that no other sperm can get in there, right? So that's how actually you can make sure that only one sperm fertilizes the egg and not 2 or 3. Because if 2 or 3 sperm were to fertilize it, then you would end up with more than a diploid organism, maybe a triploid or a quadruploid. And so, yeah, so the sperm binds the zona pellucida, releases these enzymes, no other sperm can bind. And this is where the oocyte finishes meiosis 2. So then it goes through that final round of meiosis, releases another polar body. And then the sperm releases its nucleus into the egg, into now this mature egg. And the sperm nucleus and the egg nucleus are going to find each other, and they're going to fuse together. They become pronuclei first, and then they fuse together, and they form a diploid nucleus. And so now this fertilized egg can go on and become a baby. And so now let's look at how that happens, how you go from a cell to an adult. And this is known as human development. So here we have 2 adults that are making gametes via meiosis. And each of these gametes are haploid, right? So they have half of the genetic information. And these are going to come together. The sperm is going to fertilize the egg and form a zygote. And so, yeah, there you can see that the egg and the sperm have half of the information to build a human. And the zygote is now a diploid. And so, again, remember, a diploid, di means 2. So you have 1 set of chromosomes from your mom, 1 set of chromosomes from your dad. And now this zygote contains all of the information on how to build a human. And this zygote is going to go through several rounds of cell division and become an embryo. And this embryo, these cells are going to continue to divide and differentiate, which simply means they start becoming specific type of cells like skin cells or neural cells. And I just really want to make sure you feel comfortable, make sure that you feel comfortable with what gametes are, what zygotes are, and what embryos are. These are keywords you should be familiar with. And so this embryo is going to continue dividing, and amazingly, these cells know exactly where to go and how to grow and how to differentiate, and you begin forming different tissues and different organs, different regions of the fetus. And this fetus is going to continue to grow and become a baby through more mitosis and differentiation. And right, this baby is going to be born and then continue to grow, become a child, and then an adult. And you can see there that mitosis is happening ever since you are a fertilized zygote to the time you're an adult. So this is how you go from 1 cell to 37 trillion cells. And so, yeah, you go from gametes that fuse and form a zygote that then divides and becomes an embryo, and that continues to grow and become a fetus, and then a baby, a child, and an adult. And that's human development. Very brief. Okay, so let's bring a few of the concepts that we've learned together, meiosis and gamete production, and apply it to this question. So in Turner syndrome, a female is born with only one X chromosome. This disorder could arise as a result of non-disjunction in spermatogenesis, oogenesis, or either one. So let's break this down. A normal female has two X chromosomes, right? And a male has an X and Y chromosome. We haven't actually gone over that, but even through pop culture, you've most likely heard that before. So a female with one X chromosome is not normal. And this is actually a disorder caused by non-disjunction. And remember, non-disjunction is a error in meiosis where you don't separate, where the homologous chromosomes don't separate in anaphase. They stay together. And this can cause trisomy 21, which is Down syndrome, or it can cause Turner syndrome, where you only have one X chromosome, and it actually can cause several or other genetic disorders. So this is asking you, though, is this female with only one X chromosome? Would this happen during spermatogenesis or oogenesis? And the answer is either one. So non-disjunction can produce a gamete with two sets of one chromosome. So for example, instead of having only one chromosome 21, you end up with two chromosomes 21 in, say, a sperm or an egg. And then when those combine, you end up with three chromosome 21s, right? That's what happens in Down syndrome and trisomy 21. And you can look at that slide that was in our last lecture. If you have no idea what I'm talking about, I highly suggest you go back and look at that and just look at what non-disjunction is. So basically, in non-disjunction, you can produce a gamete with either, instead of having only one of a certain chromosome, you have two, and then the other cell it can produce is it has none of that certain chromosome. So in this case, in Turner syndrome, what's happening is that either the egg has gone through non-disjunction and has no X chromosome, and then the sperm comes along with an X chromosome and fertilizes it. So that's how you can end up with one X chromosome if the non-disjunction happened in the egg, or non-disjunction can happen in the sperm, and a sperm that has no X chromosome but has every other chromosome can fertilize the egg, and the egg would have an X chromosome. So it's hard to explain without the visuals. I apologize for that. But if you go back to our last lecture and go through non-disjunction, this will make sense. The question, though, here that's asking is tying together spermatogenesis and oogenesis to this error in meiosis. And then I highly suggest that you watch this video. It is absolutely beautiful, gorgeous, graphic 3D animation of going from a unfertilized egg to a baby, and I think it even animates the birth in a really interesting way. But very, very cool video. It's a little lengthy, I think like 10 minutes, but worth watching. We might even watch some of that in class because it's beautiful. Okay, so now let's actually get a head start on inheritance. That way, on Tuesday, we can all be a little bit caught up. And this is really going to be covering some of the same stuff that we've already looked at. So it shouldn't be too much on your plates. Okay, so let's start with this guy. This guy is Gregor Mendel. He's considered the father of modern genetics. He was actually an Augustinian friar, basically a Catholic monk, and spent 10 years studying pea plants. Ended up studying somewhere around 30,000 different pea plants. Doing experiments and looking, he was curious, right? We saw that science starts with curiosity. He was curious at how these pea plants could pass on certain traits. So he noticed that there were certain traits in the pea plant that were either one or the other. So for example, seed shape, he noticed that they either had round seed shapes or wrinkled seed shapes, or yellow seed color and green seed color, or purple flowers, white flowers, or they had tall stems and dwarf stems. So he looked at these traits and he studied these plants over a long period of time and started making observations about traits and how traits are passed on. And what he ended up proposing was that traits can be inherited, so they can be passed on. He ended up proposing that traits can be either recessive or dominant, that pairs of inherited factors, which he didn't know were genes, but we know now are genes, that pair of inherited factors are separated during gamete formation. This is called the law of segregation, and we actually know that this happens during anaphase of meiosis. So anaphase separates the two chromosomes, right? The one from mom and the one from dad. And so they're actually, these factors are inherited independently, are segregated, basically, are separated. And then he proposed that inherited factors are inherited independently. And this is the law of independent assortment. So basically that the seed shape and the seed color, they don't always get inherited together. They can be inherited independently, right? So, and this is explained by the law of assortment because these traits are on chromosomes which can be assorted differently depending on how they align during metaphase. Again, if none of this is making sense, really suggest you go through the slides from the last lecture and even our last online lecture where we talk about crossing over and independent assortment. And so this was before anyone knew about genes. That's what the crazy part is, right? He was making these observations about traits and inherited factors, and we had no idea about genes. And we later discovered that these inherited factors were actually genes. And his proposals were so spot on that they still hold true to this day. So this man was brilliant, brilliant guy. And we owe a lot of what we know about genetics thanks to him because he was way ahead of his time. So let's look at this law of independent assortment. And this is essentially what we've already looked at in meiosis. It's just that now we're going to look at it as far as how genes are passed on, right? Okay, so all of the cells in your body have 2 copies of each chromosome. That is 2 full sets of instruction manual pages, 1 from each parent. So now the analogy is an instruction manual. And you have 2 full sets of instruction manuals. And it has 23 pages, right, that are like your 23 chromosomes. So sperm and egg cells each have only 1 full set. They're haploid, right? They have half the amount that normal cells do. And these sets are a random combination of maternal and paternal instruction pages. So here you can see possible sperm cell 1 has page 3 from dad and page 9 from mom, whereas this other possible sperm cell has page 3 from mom and page 9 from dad. So this is what we've seen during independent assortment. So what independent assortment does is it shuffles chromosomes. And so it shuffles the way they align during metaphase. So you could end up with a mix of your mom and your dad's chromosomes, or the pages that make up the instruction manual, the 23 pages. So every round of meiosis will always give rise to haploid cells that have a different combination of your 23 pairs of chromosomes. And because of that, that generates a ton of genetic diversity. What it ends up being is over 8 million possible combinations for each gamete. 8 million possible combinations of chromosome 1 from your mom and dad, chromosome 2 from your mom and dad for all 23 chromosomes, right? That's why it's 2 possible combinations for 23 chromosomes. It's 2 to the power of 23. And the crazy thing So what the 8 million possible combinations of chromosomes mean is that you could make 8 million gametes and no two would be the same, right? No two would have the same combination of chromosome 1 through 23 from mom and dad, from your mom and dad. So right now we're just talking about making gametes, either sperm or egg. And what's amazing is that once you get to the point of fertilization, then you increase the possibilities even more. So once an egg and a sperm fuse, that increases the possible combinations of chromosomes to 70 trillion. So that's because you have 2 to the 23 possible combinations of chromosomes in the sperm, 2 to the 23 possible combinations of chromosomes in the egg, and together that's over 70 trillion possible combinations, 70 trillion zygotes, possible zygotes. So not only is it that your mom and dad could make 8 million gametes and they'd all be different. They could actually come together though and have 70 trillion children and they would all look different. And we're not even talking about crossing over here. That adds another level of complexity. This is just independent assortment and fertilization. So it's absolutely mind-boggling. It's a powerful way to increase genetic diversity. So now let's look at how these two shuffled manuals can come together and make a diploid zygote. So you inherit 23 chromosomes or pages of the instruction manual from your mom and 23 chromosomes from your dad. So basically they're the same chromosomes. They're chromosomes 1 through 23. They're the same pages of the instruction manual, but they might have some different words here and there. They might have different versions of the same genes, right? So like for example, your mom, the mom could be contributing a brown eye color gene and the dad could be contributing a blue eye color gene. But they're the same gene, just maybe different versions. So the way that this analogy in your textbook runs is like, for example, say page 1 is the page for eye color, right? And so both the egg and the sperm have page 1, but maybe the page 1 in the egg is going to be a brown version, or the instructions of how to make a brown eye color, and page 1 in the sperm is going to be instructions of how to make blue eye color, for example. So those two manuals, they're complete manuals, right? Page 1 through 23 can come together and now make a zygote, which is going to be basically 2 copies of the instruction manual. So you're going to now have 26 pages, right? Or sorry, 46 pages, which is equivalent to 46 chromosomes. And again, I'm just going to reiterate this again and again just to make it clear. It's not that you have now pages 1 through 46. It's now that you have 2 copies of pages 1 through 23. And again, this new zygote may have 2 different alleles for each gene, one from each parent. So let's look at alleles a little more closely. So in this analogy, each line of a manual page may contain many words or genes. So for example, here you see this line from page 1 that has all of these words: build, grow, long, dark, and each of these words are genes, right? And certain genes are expressed only in specific cells. And remember, we talked about how expressed, what we mean there is when the gene produces the protein or is used to make the protein. So for example, when the gene for RNA polymerase, which is that enzyme that is involved in transcription, when that gene is expressed, it means that that gene was used to make that protein, RNA polymerase. So certain genes are expressed only in specific cells. You can see that the gene build is both in muscle cell and in the eye cell, but then the muscle cell has genes that are only expressed in the muscle, like strong heart and muscle. And the eye cell has genes that are only expressed in the eye cell, like dark brown eyes, right? So you can have two different alleles or versions of each gene, one from your mom and one from your dad, right? So you can inherit one version of eye color from your mom and one version of eye color from your dad, but it's still the same gene. And so these alleles arise by mutations. So let's look real quick at the different kinds of mutations that you can have. And here what's happening is that you're copying one of the original pages of the instruction manual onto another, right? So this is analogous to replication. And during that process, sometimes you can randomly acquire a mutation. So DNA polymerase, which is the enzyme involved in replication, can sometimes make a mistake. So in this case, this is a mistake that doesn't affect the protein. So here the original version of the gene is spelled gray with an E, and you just by random chance introduce a mutation and you change that E for an A. And now the mutant allele that you've generated is gray, right? So these kind of mutations don't affect the protein. So the protein can still function the way it's supposed to function, right? In this analogy, the word still has the same meaning. But there are other mutations that you can introduce where it changes the meaning of a word or it changes the function of the protein that the gene is coding for, right? Because remember, right now we're just talking about the gene and the DNA sequence, which is going to be used to make a protein. So in this example, the original allele is strong, spelled with an O, and when you replicate, you introduce a mutation, and now it spells string, right? So you've replaced that O with an I by random chance, but this mutation actually changes the meaning of the word, or it changes the function of the protein that's going to be made using this gene. And so this is how, in a similar fashion, a mutation can actually can create a protein with a new function or with a different function than the original protein. And then there are other mutations. These are mutations that can actually destroy the meaning or the function of a protein, right? These would be bad mutations. These are the mutations that generally result in diseases and disorders. So in this case, the original allele is nerve, and you introduce a mutation that changes the E for a Z, and now you actually have produced a new allele, a mutant allele with no meaning. So that's analogous to you introduce a mutation where now the protein, an amino acid has been replaced, and that destroys the function of the protein, and it can't do what it originally has done, right? And that would be a dysfunctional protein that might lead to a certain disease. So this is just simply how these mutations, random mutations during DNA replication, can affect genes and therefore the proteins that they code. And this also contributes to genetic diversity. So remember, mutations are actually the primary source of genetic diversity. They're where genetic diversity first happens. And then we've also seen how you can get genetic diversity in meiosis when you go through crossing over and independent assortment. And then you can get genetic diversity in sexual reproduction when you bring two gametes together and you increase the possibilities of different organisms. All right, and that is the end of this lecture. This is this first part of the inheritance was to get your mind working on this stuff, and we're actually going to go a little deeper next week or the week after spring break. So if you haven't gone on spring break yet and you're listening to this, have an awesome spring break. I hope it's super enjoyable and restful. And if you're getting back from spring break and listening to this, I hope that you had a great one. So see you guys on March 17th. for our next lecture. Peace.